We're going to go ahead and start the third portion of the symposium. This will be presented by Ms. Piper Schwenke. So talk about the marine forensic applications using high-throughput DNA sequencing. Again, like the first two portions, feel free to ask any questions that you may have on that. And at the end of her portion, what I'd like to be able to do is open it up for questions for any of the three presenters that might have come up that you'd like to maybe ask some questions. And then after that, we'll do the post-test. But the presenters will be around here for a while longer. So even after it's done, if you don't feel comfortable asking that in a group setting here, you could speak to them individually and ask some questions. If you have cards on that, I would recommend that you exchange that. Who knows, that could be a source of information for you in the future on that. So as I said earlier, for the Texas peace officers that are in here taking this is eligible for TCOL credit. What I'm going to ask is, I'm going to hold this packet, as I said, as hostage, right, until I get my exit surveys and pre and post test on that. But once you're completed with that stuff, if you see, it would see Miss Monica Diaz here. She'll be outside. She'll have the packets for you, completed, signed, things of that nature. And she'll give that to you so you can give that to your appropriate training folks. So, thank you.
All right, you've all stayed for the end. Thank you. Well, first I want to thank Victor for inviting me to come to the symposium. This is really interesting. I've really enjoyed hearing the talks of Jennifer and Karen. So, but I want to show you a picture of where I'm coming from, not very far, but this is Seattle, Washington. This is the Northwest Fisheries Science Center. The location is right on the water across from the University of Washington. So lots of great resources there. The center is very diverse. This building here, I'll circle with my mouse, was the original Bureau of Fisheries that opened in 1909, I believe. And so the building is built on since then to house more of the more modern science that goes on there. So everything from environmental toxin research, harmful algal blooms, the Gulf oil spill samples were analyzed there and the impacts for the marine life. And me in conservation biology. So today's, I just wanted to outline a little bit. So it won't jump around a lot, but they'll go through some different topics, including introducing how high-throughput DNA can be used in criminal wildlife and fish investigations, and how we assign evidence to geographic origin. I have some case studies that I've worked on that involve using high-throughput DNA techniques to find out what a sample was or was from. And then also, since I do all this in a forensic setting, I end up going to court, or will eventually go to court if anything works out in my favor for my training, to talk about the results I have in court. So I have a slide that talks about some of that. And then if there's something while I'm presenting, I'm not bothered at all if someone wanted to interrupt to get clarification or had something they wanted to ask during this presentation. I want to acknowledge the forensic team that works at the Genetics and Evolution program at the Northwest Fisheries Science Center. Trey Knott, Kathy Moore, and I are the forensic scientists that do the work in marine organisms, and our supervisor is Linda Park. This slide shows some of the cute alive versions of some of the organisms we work on. This next slide shows how they're not the not so cute and dead versions of some of the organisms we work on. The variety of forensic evidence that we get in illegal crime investigations is pretty vast, from boots made out of endangered sea turtle, endangered bird feathers, shark finning, rhino horn, illegal timber, and caviar. And also it goes in for seafood. There's container filled with seafood that is, they're investigating whether it's been mislabeled, that it's illegal product. In these investigations, mostly they're interested in finding out what species are we looking at, what species of bird or crab or sturgeon. So unlike human forensic investigations where they're trying to link the evidence to the crime scene or to a suspect, in our investigations we're actually trying to investigate whether a crime actually was committed. So the community of practitioners I work with, I'll call just in general wildlife forensic science. It's a diverse group of people working in morphology, pathology, chemistry, and genetics, and work on everything from plants to fish. I didn't list that plants and microbial research would also fit under this kind of umbrella, because we're all kind of working in this non-human forensic research. When I was part of this Society of Wildlife Forensic Science that we formed in 2009, it was to basically come up with standards, schemes for certification for practitioners and for educating those of us that are doing this discipline. And we thought, well, why don't we just be just encompassing? It's called non-human forensic science, but that actually kind of starts to feel like we're not humans doing the science. So we left it at wildlife. I just want to say that the society is open to all. I thought I was even mentioning to Jen and Karen that this might be something, if they continue on this path, it's a good resource. And there's people from all over the world that are part of the society. The next meeting is in Edinburgh. The monetary value of crimes in international trade with fish and wildlife is significant. Taken together, wildlife trade and IUU fishing, which is illegal, unreported, and unregulated fishing, and illegal timber, is estimated to have a value of over $50 billion a year. Taken together, these three components have a value, the third most expensive crime after drugs and weapons. For IUU fishing, which our group has kind of been more pulled into, there's a President Commission on IUU fishing. There's some reports out there that estimate that 20% of the seafood in trade is actually from illegal, unreported, and unregulated fishing. And this is just back to the practitioners. We basically have formed a working group to kind of bring some of these standards to the rest of the forensic community. They do forensics and chemistry, and so we wanted to find a way to fit within that, because if down the road they start to mandate certain accreditation and certification requirements for forensic practitioners that work in government, which is me, we wanted to make known that there's a lot of caveats when working with wildlife. For example, in human DNA labs, they're working with one species. We're working with thousands of different species. This is now part of National Institute of Science and Technology. These scientific area working groups are now under one government form, and so that we are able to standardize with the human forensic world more. So these are my clients: NOAA Fisheries, Office of Law Enforcement. It's there's areas on the map. You can see how regionally dispersed they are from the East Coast, Guam, Pacific States, and Alaska. And these are a list of some of the laws that they are tasked with investigating and enforcing. So the Endangered Species Act, the Lacey Act, Marine Mammal Protection Act, and Magnuson-Stevens. I think in this next slide I'm going to go over more with what are the majority of the casework that I work on is endangered species and Lacey Act amendment cases. So I just realized this slide, the numbers of special agents I have here is 130. This is old data. There was we've lost, I think, almost a third of our special agents in NOAA over the last 4 or 5 years for various reasons. So we're trying to rebuild the amount for law enforcement in NOAA. So for ESA, or endangered species, so this is just, there's a list. People know what ESA when we talk about lots of critters, but in the fisheries world, we have, there's a little bit more to the ESA than just it's the species. So certain populations within the species are more in danger of extinction than the species of the whole. For example, salmon. There's salmon in parts of the Columbia River that are critically endangered, while the species as a whole, the Chinook salmon, aren't. So they actually are now able to list certain populations or distinct population segments of certain vertebrates so that they can get protection. So that's most of the world I'm working on with ESA, endangered fish. But the Lacey Act is also really important, also criminal investigations for Lacey Act, in two ways. First of them is it's illegal to take and process and transport between states illegal products, wildlife and fish. And you probably know CITES is more the international laws that prevent that, and the U.S. Fish and Wildlife works more with that. That's the ivory and the tigers and a lot of those international imports of illegal. There's a list on CITES. But also Lacey Act could be an investigation if you were working on mislabeling. If you're labeling a product as, what was your example, Jen, where you're as?
Oh, it was the tuna and the tilapia.
Oh yeah, tuna and tilapia. If you're calling tilapia tuna, yeah, that's definitely a Lacey Act violation. The laboratory I work in, although I joined in 2000, NOAA has been offering forensic expertise to OLE back to the 80s. They were using proteins to identify fish in high seas fishery investigations. On our side, it's tuna, and in the Atlantic, we're at salmon, and the East Coast was more tuna. But as technology improved and the DNA became online, we are doing mostly DNA testing for law enforcement. To set up a forensics laboratory, the two main things is that you need to secure the evidence, and you have to have facilities that are dedicated to processing evidence. The other thing is that if the scientist is willing to go to court and has the credentials to do this work. So bringing it to the topic of today's symposium, I wanted to just, again, reiterate some of the differences, the technology that we're using during these wildlife and criminal investigations. So for metabarcoding versus metagenomics. So the most of what I do is genomics. I'm looking at population genomics. So I thought it would just kind of segue what we learned about metabarcoding is where it's the single DNA marker versus the genomics for population, which is multiple DNA markers. And metabarcoding builds on, like Jennifer really did a good introduction of that, is that we're looking at a single gene and we're looking to see what it is. And to identify something using a single gene, you can use barcoding. And invertebrates, that's this CO1 gene. You can use other techniques to identify species that I'm not going to go into here, but like for PCR and sequencing is actually another way of saying barcoding. So any gene, and then also qPCR that Karen talked about. You can use qPCR real time to target the gene you're interested in and probe it for the species you're interested in. So qPCR could also be a good way to identify species. But going on to like the metagenomics, we are looking at several regions of the genome and genes that are different between populations of a single species. And traditionally, this was done with microsatellite markers. Microsatellite markers are the one we see in the DNA fingerprinting in the human forensics labs. It's 13 loci that are used for matching. We also use those type of markers to look for population differences in the species range. We also use single nucleotide polymorphism. So this is just another technique or another method for looking at differences across the population, across the range of the species. And I think here was a good point to mention is that when we talked about SNPs, when we're looking at those short strands of sequencing that come off, those high throughput sequencing, those have SNPs in them. So we're actually looking at the single nucleotide polymorphisms in those short sequence reads that come off those instruments. I didn't put any pictures of machines. They did. Good. So I'm using those machines too. To kind of put it into context, I was beginning to think of maybe some good examples of how these methods could be used in a forensic setting, though they haven't been yet. So for metabarcoding, so looking at finding a species in a mixture, like what are we seeing? My screen's a little bit difficult. Sharks. Oh, sharks, yes. So the collagen elastin fibers found in shark fin soup, which is the end result of why there's all this shark finning, is basically looks like this. This is what goes into the soup. There's no morphological characteristics that can identify it there. This is my colleague Jenny Giles, who came to Seattle as a postdoc. She's interested in the species variety of sharks in the shark finning trade. And what is there, over 400 species of sharks? So it's a huge diversity. And this, I believe, would be a really good, in fact, she wrote a grant and is now at Stanford. She's going to investigate using metabarcoding to look at mixtures of species in, let's say, a sample of this ceratotrachea, this shark fin soup mixture. Because at this point, here's her looking at piles of shark fin in Seattle after a seizure at the Seattle port. This is just piles that she can actually, if it's in good quality, she can actually look at fins and tell which part of the shark it came from and what species it is. So here I also want to start, I think this is where I want to talk about eDNA. This is a good place. So looking at a species in a mixture, you're talking, the first, it starts with a sample. So like we're talking about pollen as a sample from the soil or from the clothes someone wears. So that's an environmental sample. People also take samples and they're looking for contents in water. And so this is what our laboratory is working with, some eDNA sampling methods in water. For a good example is that if you do an internet search, they're using eDNA to look for really endangered, hard to find critters in fresh water like the salamander. So if you can't, they're so hard to find and elusive, well, the samples of water that are taking may contain actually DNA from an animal that was there the previous day. So basically eDNA is a sampling technique to detect really small amounts of DNA in an environmental sample. So in criminal investigations for genomics, where I might be asking the question, where did something come from? In my world with fisheries, the fish in the ocean look really similar. So fish from Russia might look really similar from the fish from Alaska. But some of the interest in illegal imports is, did it come from Russia? Well, they're right there. They're almost touching in the oceans of the Bering Sea. So we are interested, and a lot of scientists are interested in looking at as many DNA markers as we possibly can to actually see if we can tease out some of those differences to look at illegal imports in king crab, pollock, and these are really widely distributed fish stocks that are commercially important. And also in the European, see these icons that I put up here, Fish Pop Trace and Aqua Trace, are actually doing this in the Atlantic. And they've got some really good funding here from the European Commission that we haven't been able to figure out how to get on the US side to actually develop these baselines of their important economic fish that are in trade and are also that are in hatcheries. So knowing if something came from a hatchery, they're actually trying to get baselines from all the hatcheries so that in the market, you know that it's been properly labeled and then what hatchery it came from. Various other applications. Okay, this is my slide to remind me that we're talking about some case samples. So here's a bunch of abalone. And I wish I had the value of this box right here with the pen in it. I had 4 or 5 boxes of these, and these are the mussels from the single, it's a mollusk that has single shell. It's beautiful shells. But once you remove the shell from the meat, you don't know what species it is. And so this was a seizure that was coming from Mexico across the border into the US on its way to Canada. This here, the heart-shaped little guys, came from another seizure from near when I first started at NOAA. And this is before my methods were really developed in abalone. There's 8 species of this abalone on the coast of the eastern Pacific Ocean. So these little bars show kind of the range. White abalone here is critically endangered in the US, and it's listed on the Endangered Species Act. Also black abalone, whose range is also kind of in the south. Probably can't see it because it's black. Here it is, against the blue background. That's also listed on the Endangered Species Act. But what's going on now is they're really interested in this other species called Pinto, right here, Pinto. And its distribution, actually, the species complex is distributed from Alaska all the way down to Baja. But in the southern part of the range, a lot believe that it's a subspecies, it's different, and they call it threaded. But Pinto in the north, which I think I have a slide that talks about this more. The Pinto in the north are listed in Canada under the COSEWIC as, that's their Endangered Species Act, as threatened, and there's no take on Pinto abalone. But it's very plentiful in the south, and it's called threaded in the south. So if we're looking at trying to protect the species, that's the question for the manager. The manager one day might want to list Pinto abalone in the US. In fact, it's on the list to be considered. There's been, NOAA's been petitioned a few times to list it. But I think the question for us is, well, can we even tell if it is the endangered subspecies? Is it different than the threaded, or is it just diversity of species across the range? Here's a couple of pictures on here. These were actually from a case where they were importing dried abalone, and there is black and white abalone in this seizure. Here's the threaded over here. They're less commercially important, but ecologically they're very important. They're smaller, though I wouldn't be able to identify these without molecular markers. So I set out to figure out if we can actually find a difference. Molecular genetic tools at this point cannot tell the two different between the Kamchatkana and the Assimilus, so the northern Pinto and the southern threaded. And it's at this point, I'm just looking at these pictures, they're just beautiful. But unless I worked in fisheries, I probably wouldn't know much about the history and the life history and the ecology of these fantastic organisms. So I set out to, before, I used to picture some shells, same thing. The shells, there's also some ambiguity with the morphology of the shells. So even these shells, which come from, they believed, known individuals of Pinto and threaded, it's even difficult to tell with the shell morphology. No genetic difference between the two found. For species ID, I actually used two markers. I use a barcoding marker, the CO1. I also use a nuclear marker that was isolated out of gametes of this broadcast spawner, and it's very, very specific to species. It's been extremely helpful in identifying abalone. But still, even with that marker, I'm unable to tell the difference between these two. So I set out to do a RadSeq experiment. And this slide I'll kind of walk you through it. It's just basically how I set up this experiment to see if I could find these differences. So the purpose of the study was to look for SNPs, or differences between these two subspecies. I used the method of RadTag sequencing, and what I'm looking to is sequence as much of the genome of these two as I can and look for differences. I also added some other species in there that are closely related, because in the event that I find a lot of different variable markers, maybe I can find a new species marker for me to do more high throughput barcoding of these guys when I get them as a case. So this results section shows you is that my yield of it wasn't great. I only got 100,000 sequencing reads. I guess that doesn't sound too bad, but the usable ones were very low. So I only got about 1,200 sequences that are variable in my samples. But when I got drilled down to it and I started targeting these areas, in fact, I found that I could find no differences between the two. There were some candidate areas that might prove for future research to look for. So that was the end of that study. Eulachon is an anadromous smelt in the Pacific Ocean. This is really interesting. There's a scientist I worked for that has done some historical research for this. It's a very important cultural fish in the Northwest. It's a smelt about this big. Some of the cultural references for this fish are that the natives in the area used to call it the salvation fish because it came back in February and March, when everything else, all the salmon was depleted and all the other meat was depleted. So this is the salvation fish. It is full of oil, and you actually can dry it and burn it like that. It was an important trade feature. So the native tribes in the Northwest used it for trade. They carved these intricate vessels, and they rendered the fish down just for the oil, and they call it ooligan grease. The picture up here shows the ooligan grease. Can't imagine what a vat of rendering probably smelled like. But once they got it down to this, it was like grease gold, and they were able to it was their prize for trade. So as even Lewis and Clark, when they first came through, found it very interesting. They Lewis wrote this journal article here with a little sketch of a eulachon, and this quote at the bottom is from this journal, saying that I think them superior to any fish I've ever tasted. And most of us, I don't know if anyone eats smelt, like little fried up fish. I want to taste it now. Before, I think I was not interested in it. So there was a fishery for it for many years. So not only the natives with their tribal rights for the fishing, but also the Northwest for food fish. And it was very popular, and they were plentiful. It was like the salmon story. You could walk across their backs when they were returning. And but in 1995, the fishery crashed. They weren't coming back to the Columbia River. and of course NOAA was investigating how this was happening. I think they quickly listed it. I think it probably took a while. I think it was in 2010 they actually listed it as an endangered fish. But like I mentioned in the previous slide about population segments, the whole species range goes across the Bering and into Russia. So it goes to California historically. So they did a bunch of genetic research and found that only eulachon that are from part of BC and in Washington are the ones that are critically endangered, and those are the ones listed as candidates. Or they became listed under the Endangered Species Act. And so my question is, when I get a pile of evidence of eulachon and showed in these pictures, is that's a geographic question. Were these from our local population of eulachon, or are these something from Alaska or imported? From what we know about eulachon, the distribution, as I mentioned, goes across the top here of the North Pacific. And here in this bracketed area shows the range that was found to be that was listed as threatened under the Endangered Species Act in 2010. So in fact, this actually spanned across the U.S.-Canada border. That's kind of interesting. Usually these ESA listings stop right at the border, which, the fish don't see the borders. Lucky for me, the researchers in fisheries were very interested in this as well, and so they published a baseline. They used RadTag for this baseline, so they actually used the method, and they interrogated the whole genome of several populations of these fish across their range, but it's not complete. Actually, I realized that it stops here in Alaska, though the fish do continue across the Bering Sea. So they have a sample here from Alaska, one from BC, and then one from the Columbia River and the Fraser River down here in the Salish Sea area, which is in Washington and BC. And the data showed that over 3,900 SNPs, these markers, showed a big improvement over the previous method used to look at population differences between the fish, which was the microsatellites. Here's a scatter plot here on the right that shows the different populations, how they fell out when you look at differences, which is pretty good. So these are the Columbia River, these are Alaska, and these are kind of the BC. So I thought this was a promise. A law enforcement investigator was looking, I showed you fish earlier, that can we tell, this retailer says that these fish weren't caught in this fishery. We know this fishery was closed. We know it's illegal to take them at this time. We believe their defense is going to be, they came from Alaska. My buddy sent them down from Alaska. So to kind of preempt that defense, we just wanted to we wanted to look at the DNA and see if the fish that were collected as evidence actually came from our local stock that's listed. So I used the same methods that they used in the paper. I have now a whole bunch of sequencing data that I got 2 days ago, and I haven't been able to look at it. So the question remains unanswered of whether this method can be used kind of for prime time, as I say, in a forensics method. So basically I'm validating it with known samples, and then I'm going to test the evidence and see if I can assign them to one of these colored blobs using population genetic techniques. So speaking of forensics, this is one very busy slide, but it's very dry stuff because this is all when I go to the law seminars of the attorney, or there's probably attorneys in here maybe, but their slides are usually pretty dry. But this is really important stuff because especially when you're presenting a new method in court, it's called a Dalbert hearing or FRI, and this is basically asking about the science. Here's the questions that they're asking: is this been tested? Are there standards that control this technique? I mean, how do you know you don't get the wrong answer? And they want to know if this technique's been peer reviewed. Is it been published? And then this is the hardest thing for a lot of forensic science here, is knowing the potential error rate. And so going into courts now, I was being prepped for a trial for an endangered salmon case, and we were sequencing, and the question was, so you need to quantify your potential error rates and be able to present that in court. And that was a very difficult question. We need to know the error rate, but was this human error rate, how well I do, or is this, the technique has intrinsic error associated with it? So before I go to court on a sequencing case or any analytical case, we need to at least know what the error rate is. And it's never, you can't say it's 0. You can't say you're perfect, apparently. General acceptance to the theory, and then you have to consider the alternative explanation. That's going to be an important thing too. Yeah, and so I think they're just trying to say your conclusions based on real data. Were you able actually to find, does this make sense? And can you quantify it? And then what was the error rate? There we go. I'm done. Thank you.